Okay, so today is the last class where we are going to talk about DNA synthesis. In the previous class, we talked about DNA sequencing, how to read DNA, and I gave you an overview. Of course, as I mentioned, for DNA sequencing also, there is a possibility of about 10 lectures because each sequencing method you can describe in detail in chemistry. So you have to go into your chemistry background, right? Same thing happens with DNA synthesis also. But as we are not going into too much technical details of chemistry, but I will give you an overview, right? What is there going on in the DNA synthesis? And, and we hope certainly because of the technology like DNA storage, there more and more progress is happening in one of the key, approach of DNA synthesis. So this is really very, very interesting. And since because of the Human Genome Project, most of the effort was there on DNA sequencing. So we see a technology just like nanopore sequencing, which is very small. On the other hand, not much work has been done on DNA synthesis. Of course, there was a one popular method discovered by Marvin. Marvin, he has not received Nobel Prize yet for this discovery, but still, it's an important work that he has done. And in fact, Marvin and I, we both share same date of birth, right? So, which is good thing. Anyway, so he has discovered this technique something around 1950s. So I'm going to give you some brief historical overview and how things evolved and what are the key techniques that we are using it. So it will not take much time because anyway I'm not going into many too much technical details because these are if you really want to go, there are plenty of nice things about biochemistry, and you will see in the even the journey of DNA synthesis, each and every step people have done something and they have received Nobel Prize. So you still have an opportunity, right, if you can focus on some of these things. So we talked about codecs. Of course, we have done only a couple of codecs, and there are plenty of them, which are there. I have not talked about storage and retrieval, which is kind of mechanical things. But anyway, sequencing we talked about. Now I'm going to focusing on DNA synthesis in this process. And most of the slides are based on this review paper which appeared in 2014. So again by George Church Group. So same name you can take anywhere, from discovery of this field to all the major things that he's doing. And in fact, this is the only review in the last how many years? So 2014 is about 8 years or something. Since then there is no review, so it's an opportunity for somebody to write a review on DNA synthesis. So many things have happened after that. But anyway, so this paper is interesting one. So there are three kind of DNA synthesis. One is in vivo DNA synthesis. In vivo means which is occurring in nature already. So nature does DNA synthesis, right? So this process was observed, of course, by many people, and I mean mostly this is around 50s, 40s, 50s during that time, where people were observing DNA synthesis. Chemical DNA synthesis started in 1955 or so, and recently, since last couple of years, enzyme-based DNA synthesis is becoming popular. And it is also known as green DNA synthesis. So green means no waste, not much toxic things that are produced here. So I think these are the three major approaches. Now, depending upon how do you implement the technology, there are column-based, so column synthesized oligos, right? Array-based gene synthesis, and so how do you do? Basically, one way is you do just bit by bit. Another way is you do it parallelly. So using some sort of a microarray kind of technique and so on. So this picture is showing what are the commercially things available and the cost per 1,000 bases. There are a couple of companies in India who can synthesize DNA for you, right? So like one of them is Genome Patri, I told you earlier. So in Hyderabad company started by Anu Acharya. So I think their cost is 15 paisa per base pair, for designing the DNA stuff. Okay. But so this is the whole technology bottleneck is DNA synthesis. As soon as more progresses will happen here, technology will be commercially available, I think within 5 to 10 years, I think. So this is an important area. So I'm going to talk about chemical DNA synthesis, how this is started. So Alexander Todd, he's a British Nobel laureate, he got, I think, Nobel Prize in 1957. So he started work on vitamin B1, all these biochemistry people in 1934. And actually in 1950s, he started work on vitamin B12. Okay. So while doing that, he observed that you can synthesize, this DNA through some artificial means as well, block by block. So one bit by one bit, like, so basically, in fact, he got this Nobel Prize in chemistry for his work in the synthesis of nucleotides, and actually, this guy, Hargobind Khurana, you must have heard about his name. He born in Punjab, right? Multan, I think, and then in 40s, 47, 48, I think he finished his PhD from England. I forgot the university name, but then he came back to India in 1949 and he was looking for some opportunity. Then he went back and worked with basically, Alexander Todd with him. And there he learned how to synthesize these DNA things, initially. But he got Nobel Prize for some other thing. So, he got Nobel Prize in 1968 for the interpretation of the genetic codons function in proteins. So that map from DNA to RNA and RNA to protein, right? So how that translates. So which codons will give you what kind of protein sequence, protein alphabet. Those discoveries were done by Hargobind Khurana. So very famous name, very respected name in the biochemistry area. Okay. And he also designed the world's first synthetic gene. So, also he did that work in 1970. So we can say you can call him father of chemical biology, like the person who really started these things at a very, very, early time, So he died in 2011. By the way, there is a paper he wrote in 2012, of course, before his death, which appeared in one Indian magazine. So there you can find detailed history of each and everything that was created in DNA synthesis, how he arrived at different ideas and so on. That is discovered. In fact, he covered a lot of other interesting aspects of DNA synthesis. So this is so basically from Todd to Hargobind Khurana, right? And then comes Marvin, So Marvin has, basically he is known as actually main person for the chemical synthesis of DNA. And this paper I have taken appeared in 2013. Chemical Synthesis of DNA, RNA, Our Gift to Science. Nice title, right? How, so I have taken a couple of pictures from this paper itself to tell you what was done. So actually, Marvin did his postdoc with Hargobind Khurana. After PhD, he was working with him, and that's where he picked up these things. So last person is Hargobind Khurana, and this is Robert Letkin. He was the basically so Bob was the advisor for Marvin, so he was working with him, and I think at the time when they were doing these things, so he was also trying to Bob was trying to synthesize DNA, and actually there was another person who got Nobel Prize again for the same work, similar work basically, and Marvin was thinking that probably Bob will also get this. Anyway, so this is something. He's a Marvin. So now he is credited for discovery of solid phase phosphoramidite chemistry. So basically, this is the idea behind chemical DNA synthesis, and this was automated also. So basically, you can try to automate the thing. So DNA synthesizer can be automated. So this technique is very, very popular until the, still there are many companies which are working on this technology, and they are producing DNA. For a lot of other things, people are using this technology. So basically, here is this chemistry. There are 4 cycles, this technology produces 30 to 120 nucleotides. Okay? So you can synthesize 30 bits to 120 bits of ACGT. And it synthesizes 96 to 384 oligos simultaneously, How we do this, what is DMTO, what are all the B stands for bases, ACGT. So we can go into chemistry, but I'm not going into chemistry, right? But if you want to look at more details about this, and if you have interest in chemistry, I'm sure you will have interest in chemistry because chemistry is all computer science. So you will find that, so there are multiple videos available where you can see all the detailed chemistry behind this process. So I'm not going into the detail, but at least I'm telling you, it's a 4-step process, right? For doing this chemical synthesis of DNA. And, at the time of Marvin, so they were trying to do this basically lac-operon, synthesis, they have done in the lab. And this is the whole process of that, which resulted in a whole theory for that. I mean, how it will work actually and so on. So this was the first thing that they have done. And these are 2 of his students actually. Again, this is the same 4-cycle picture, I have shown you some other source. In fact, this is the patent that Marvin has filed with his student Serge in 1983. He filed another patent. So 2 of these patents created around $45 billion industry. You see, 2 patents, great discovery in science and technology, right? So this is something interesting. These are the 2 patents which are important. And Marvin also co-founded a company, Applied Biosystems. So they have actually, produced a DNA synthesizer, the commercial DNA synthesizer. You can see that Bill is, having this, delivers the first commercial applied biosystem, 380A DNA synthesizer to the Marvin laboratory in December 1982. Okay, so all this chemical-based synthesis is still very, very, there are some limitations. So I'm going to tell you what are the limitations of this approach. But still, it was the only technology available for synthesizing DNA until recently. Okay, last couple of years, still people are using it. So these were some other people, Peter, Steve, and Doug, who were involved in this chemical DNA synthesis process. And, so what are the limitations of this, so currently it can give you up to 200 nucleotides with a 99.3% yield per synthesis cycle. Okay, so yield is important. How much DNA is created, right? That yield is important for the DNA synthesis. Okay, so but when you use longer the sequence, the lower of the final product yield. So if the sequence is longer, the product yield is less. So this is a limitation of this approach. Also, depending upon the purification method, there are 3 methods, popular methods for the purification. It could be even lower. Production yield could be even lower in this chemical synthesis. And there's a produce of toxic waste is produced in this. So it's not green. Okay, so while doing this, in fact, US government has put ban on some of the toxic waste that are generated through this DNA synthesis, obviously. And it is more error prone. So errors are more. So in all ways, though it was very important discovery, people are using it so much, industry has been created based on this, but still there are problems with this, right? And so there are some scientists who, started thinking in the other direction, like how nature produces, how nature does DNA synthesis. So they observed, and the main thing was the discovery of DNA polymerase, which has created a new area actually in the DNA synthesis. So this is known as enzyme-based DNA synthesis or green DNA synthesis because the toxic wastes are not there. This is how nature produces DNA synthesis, And these are the 5 companies: Molecular Assemblies, Evontex, ANSA Biotechnologies, DNA Script, and I think Twist Biosciences. And you can see that most of them are working on DNA storage as well because, these are the companies which are doing both, right? So this DNA synthesis obviously can be used in storage, but apart from that, there are other applications, right? You want to create or synthesize a new virus, or you want to do whatever you want to create. So, you require synthesis method, right? So this is something very, very important. But these companies are, so I think if you are looking for opportunities, you can contact these companies, right? They do hire. I think there are already about 10 companies which are working in DNA storage, right? So they are larger players, including Microsoft, who are hiring also in this area, By the way, this is a summary of DNA storage systems that has been built, and especially I have included this table because you can see that the synthesis method, so length was 115, and in the first entry, you see that it is silicon based, the chemical synthesis. Then it was microarray based, again chemical synthesis. Okay, microarray based chemical synthesis, silicon based. Okay, you can see that last 2 here are based on enzyme based synthesis. So mostly chemical synthesis is dominating in the even DNA storage area, right? So and you can see some of them are random access as well. So it's very, very early. Like for example, in this one they have included 3 JPG files, right? Using Terney code, rotating encoding, the Goldman encoding that they have used, So this is actually, now you can see that this is much better. Of course, this is based on, this paper is again by George Church. So all the landing discoveries are done by his lab, So now I was talking about DNA polymerase. So they have a long history again from 1955 to, you can see that these are the names, Paul V, Paul 4, Paul 1, and so on, And when they actively detected, confirmed, name was chosen, sequenced, and there is some controversy also sometime. Okay, from 1955 till 2015, this is 60 years of DNA polymerase. These are very, very important. Out of this family of enzymes, so these are enzymes. There are some of the 3 enzymes are very important. So these are the enzymes capable of extending 3' end of the single-stranded DNA. The first one is PNPs. So it is known as PNPs. So this was discovered by Spanish scientist Severio in 1955, and again got a Nobel Prize for this, right? So This was important thing. And in between, this is something around 70s, I guess maybe 80s, if I don't remember the exact year for this. And the third enzyme is very, very important. So this is actually is the main focus area of George Church right now. He's focusing on this enzyme, you know. So this was discovered in 1959 and was used in 1962 by Coulomb, right? And you can read. So it was discovered in cup thymus, So basically, in the mammal, it was discovered, and this can be used for synthesizing DNA. This is something. So each DNA synthesis is three-step process. Initialization, elongation, and termination. Each step, how do you control? That way, you will get the speed and other things in the DNA synthesis process. So okay. So this is basically a general picture how all these things will work, how the base is attached, and so on for the enzyme-based synthesis. Of course, it has also affected the sequencing. So this is a table for sequencing, enzyme-based sequencing. That is also another area which is coming up, and I just included the table for your completeness. Now I'm going to just say a few words about two important papers. You see again by George Church, terminator-free, template-independent enzymatic DNA synthesis for digital information storage. This appeared in 2019, based on last enzyme. That was discussed. So this is the important one. So here actually, he has shown how this will work. And he used apart from TDT, he used AP, apparase for controlling the synthesis. All these nature papers, as you know, they think about something, they demonstrate it, they do the experiment, they write the software, do everything, right? And then there is a paper in Nature anyway. Second one is little bit more better. Is photon-directed multiplex enzymatic DNA synthesis for molecular, again by George Church group. Okay? And this is in 2020. So this is a kind of parallelized enzymatic DNA synthesis. So parallelly, you are synthesizing DNA, molecules, which is an important discovery again, right? Sure, one day he will get also Nobel Prize or all the discoveries he's taking up, right? Or whatever. So here actually, he has demonstrated in this paper. So this is the last paper I'm going to talk about. This is the system that he designed. Okay, photon-directed DNA synthesis. So inactive, active, deactive, based on photon. Okay. So this is the dynamic mass, and obviously I'm not going to any details how this will work because you need to understand the chemistry also. But anyway, this is a important one, and I think they have demonstrated. Yeah, they have created a music storage thing. Demonstration of digital music data in DNA oligonucleotides, enzymatically synthesized in multiplex music data storage process. So you can see that input music sheet, encoding, the same kind of encoding, right? Ternary to ACGT, okay? And that Goldman thing, rotating the map. In vitro means, in silico means you're doing on computers, right? And then in vitro means you are doing synthesizing in the lab. So parallel synthesis of DNA is stored in, then data is stored in single stranded DNA. Reading is done through next generation sequencing, and then you decode and get the music file. Source code is available. You can look at it. I'm sure some of you might be doing the project in this. I'm not sure, right? But this is something, I think this is the latest one. Okay. Obviously, that means this is going to be the future of enzyme-based DNA synthesis. More and more work is done. So I think since this is, I would say, a couple of years old, people are working on improving the process, and there are companies like Catalog DNA, and other companies which are trying to automate the whole things. And I think Twist Biosciences also has a product which is so basically there are companies which are building the DNA printers based on this approach, enzyme-based DNA synthesis. Okay, so you can quickly write the DNA. So I think this is the whole future, I guess. Yes, so I think that's all. So we have covered a couple of things. We could not cover rewritable storage, but I think some of you are working on it. And so green storage and so on comes from DNA synthesis, which is green, right? And all those things. So I think more or less we have covered many things, but some of those things are not covered, but that's okay, I guess. So that's all I think is, thanks. So I think if you have any questions, you can ask. So I have not covered in detail about the chemistry, but that's okay. So I'm not sure if you are interested in listening to chemistry, right? But